In this lecture, we're going to review how the cell, in this case the neuron, engages and generates the bioelectrical signal. And we introduce this concept called the graded potential. And the graded potential refers to small changes in the membrane potential or the voltage difference across the cell membrane, the difference between the inside and the outside of the cell. And that's going to be generated by ion channels that open and close. Now, in order to do this, the neuron does need a stimulus. So that stimulus could be in the form of an artificial electrical stimulus, a chemical stimulus in the form of a neurotransmitter, an electromagnetic stimulus such as light when it comes to the detection of vision, and mechanical, and that's the compression of the skin as an example. So if we take a look at this graph, plotted along the y-axis is the membrane potential in units of millivolts, and this represents a change that is generated by a redistribution of ions across the cell membrane. And we see that the resting membrane potential of a neuron is approximately negative 60 millivolts inside with respect to the outside. And the x-axis here, it's not labeled, but the x-axis is time. And what we have here is a particular stimulus. And this stimulus could be the release of a neurotransmitter from a preceding axon. And so this is the response of a neuron in response to the neurotransmitter released from a preceding axon onto affecting the dendrite of this neuron. And so what we see here, the neurotransmitter is causing the resting membrane potential to move in the positive direction. So this is negative 60 resting, and this might change to negative 59, negative 58, negative 57, so on and so forth. And then it peaks, and then it comes back down to the baseline. And in this scenario, we're looking at the exact same neuron, but what we see here with the longer arrow compared to the first one is that we have a stronger stimulus, and this could be generated by more neurotransmitter released by the preceding neuron, and that's going to cause a proportional movement, greater movement towards the positive direction. And that is what we call a graded potential. And recall that this baseline here is the resting membrane potential, and the dotted line represents a threshold. So as long as this neuron potential stays below the threshold, it will return back to its resting state. Now, do all stimulations cause depolarizations? And so a depolarizing potential is what we observed on slide 28, and we see that here once again. So as this cell, as it is in its polarized state, as it moves more positive, it becomes depolarized. So depolarization refers to the movement away from the polarized state, and that's a movement towards this threshold. So the interior of the cell is actually becoming more positive. Opposite to that is what's called a hyperpolarizing potential. So we have a polarized neuron, and if it's hyperpolarized, it just becomes more polarized, and that's illustrated with this tracing here on the graph. So we start with our negative 60. We have a threshold of negative 55, and we see that this first stimulus, once again, coming from the release of a neurotransmitter from a preceding neuron, will try to move this neuron towards the threshold, but not enough neurotransmitter has generated that. So this cell will return back to its resting state. And that's what we call a depolarizing potential. It doesn't always hit threshold, but we see that it does move in that direction. In this case, here we have a stimulus on the same neuron, so the red represents the same neuron, But this stimulus is a different neurotransmitter. And when this neurotransmitter is affecting the same neuron, we see that we have a movement that makes the interior of the cell more negative, or it's hyper from its polarized state. And we call this a hyperpolarizing potential. As we move forward, think about what would cause this cell, this particular neuron here, to move from -60 in the direction towards -55. In order to become less negative, we have to add positive numbers to -60 to move it up to -55. In the cell, those positive numbers along the y-axis are generated by positively charged ions. So if we repeat this on the depolarizing side, these potentials can what's called summate or add. And the two different types of summation we can have is temporal and spatial summation. Temporal, referring to time, results in the uh, uh, movement towards the threshold as a result of an increased frequency of stimulations from one preceding neuron onto this red neuron that we have at the bottom. And with the preceding neuron being activated faster and faster, that will result in a faster and faster activation of this red neuron. The other type of summation that we'll see on the next slide is called spatial or in space summation. And that's where the red neuron here receives input from many neurons. In the temporal summation case, we're just going to focus on stimulations from just one preceding neuron. So we start with our resting membrane potential of approximately -60, which is the solid horizontal line. And once again, this is the neuron, the red neuron here, in a polarized state. And we have our threshold sitting at around -55 millivolts. So to repeat what we had in the previous slide, we have a stimulus, which is a neurotransmitter acting on this neuron. And this neuron will move from -60 to -59, maybe -58, and then we'll return back to the baseline. And then once it returns back to the baseline, we release another neurotransmitter or another stimulation from the preceding neuron, and we see we have the same effect. It doesn't come close to the threshold, but it starts to return back to baseline. Before the red neuron gets back to baseline, the preceding neuron is activated again and releases the same neurotransmitter before this neuron has a chance to completely reset. What that will cause is another depolarizing potential, and this is where the depolarizing potentials will start to summate. So the red neuron does not get a chance to completely reset, where it will just bump to a higher level. So the positive numbers or the positive ions that were added during this depolarizing phase are retained, and we have more positive ions coming in that bring it back up to the next level. As it attempts to return back to the baseline, we see that the number of stimulations from the preceding neuron increase closer in time. And as that happens, the state of the cell, in this case the red neuron, will move closer and closer and closer to the threshold. And this is how temporal summation works. In the case of spatial summation, here we see in the upper right-hand corner a depiction of a motor neuron. And to activate this motor neuron, the cell body, through its dendrites, must receive connections from other neurons. And so we highlight the cell body, which is located in the spinal cord. So once again, if we just have one preceding neuron acting on the motor neuron, this preceding neuron will act temporally and will increase the number of activations, increasing the amount of neurotransmitter released. These neurotransmitters will act on a protein receptor embedded in the membrane of this neuron and cause a depolarizing potential. But in reality, this motor neuron receives many, many inputs from many different axons. And in this illustration, we only see 3 preceding neurons. But in reality, motor neurons receive thousands of simultaneous inputs. If these are all excitatory inputs, then at the same time as each neuron is increasing its frequency of activation, the individual neurons will summate spatially. And this will have an even greater profound effect on generating what's called the depolarizing potential in this motor neuron, assuming all of these, in this case, the 3, are all releasing excitatory neurotransmitters. So this ends this video on graded potentials.